Good morning, everyone. My name is Lei Wei. I am a bioinformatician working on cancer genomics. Today, I will be talking about detection of low-frequency mutations. Next generation sequencing, or NGS, provides a convenient platform for detecting genetic variants, and our work mostly focuses on somatic mutations in cancers. Everybody here is probably familiar with tumor heterogeneity. NGS allows us to identify mutations that's only present in part of a tumor, so-called subclonal mutations. If we keep pushing the envelope and sequence very, very deep, we can identify mutations that's only present in a few percent of the cells. And this has a lot of potential utilities. The first one I'll be talking about is UV-induced DNA mutations in normal skin. The paper was published early this year. In the summertime, when we go to the beach, we are always reminded, don't forget to put on the sunscreen because sunburn can cause DNA damage and potentially skin cancers. And right now for the sunscreens, SPF or sun protection factor is mostly measured by redness, which is not a good indication of DNA damage. Can we measure the mutations in the normal tissues? The answer is yes. Indeed, in the last few years, there are multiple high-impact papers published, many of from Martin Corena and Peter Campbell's group in UK, and one of them is about the skin. So in this paper, they analyzed sun-exposed human eyelids and identify a large number of low-frequency mutations. But one key question that was not answered in the paper was which mutations are introduced by UV instead of aging or other factors. So we designed this study for non-skin cancer patients where we collected 450 skin samples from 13 donors. And these were individual matched, so half from sun-exposed areas, half from non-sun-exposed area from buttock. For sequencing, we used a targeted panel that contains 55 of 100 base pair windows, so total size 5.5 kb, and mostly in Notch 1, 2, and P53. And we did very deep sequencing. Here are the numbers of mutations we detected from each window, and red and the blue are from the sun-exposed and non-sun-exposed area. And if you look, you can see in P53-3, this window, we identified 37 mutations, and all of them were from sun-exposed skin. And similarly, there's another one from Notch 1. If you sort all the mutations by their chromosome position, you can clearly see there's a region in P53 that's never mutated in non-sun-exposed skin, but become highly mutable in sun-exposed skin. And on the mutational level, we identified 6 mutations that are exclusively mutated in sun-exposed skin. And some of these are in well-known cancer hotspots, for example, this P53 R248W. UV is known to cause C2T transition at dipyrimidine sites, so-called UV signature mutations. And our study showed only UV signature mutations, but not the non-UV signature mutations, are enriched in sun-exposed skin. And more specifically, it's only the missense and nonsense UV signature mutations are enriched. And combining the two factors, first, there are more mutations, UV signature mutations in sun-exposed skin. Second, these mutations have a higher variant allele frequency, so larger clones. And we combining these two, we developed a single score called cumulative relative clonal area, or CRCA, which is the percentage of biopsied area covered by clonal UV signature mutations. And we calculated this for all 13 patients and found that the score was higher in sun-exposed skin than the non-sun-exposed skin in all patients. And the average difference was 11-fold. We then analyzed another extended cohort from skin cancer patients. We have 8 patients with cutaneous squamous cell carcinomas, and 4 of them have at least 3 tumors, so high risk. And 4 patients had 1 single tumor, so low risk. And we collected 20 samples, including 16 normal skin and 4 tumors. And we did a deep sequencing. So the main question was if there are any difference between the high risk and the low risk group in terms of mutations in their normal skin. So here are the numbers of mutations mutations we detected from the high-risk normal skin, low-risk normal skin, and the tumors. And we realized that if we draw a line here at 1% variant allele frequency, above this line, there was not much difference. But below this line, there were significantly more mutation in the high-risk group. It's easier to see it if you turn to a histogram. Where you can clearly see this elevated mutation burden below 1%, and it was statistically significant. I'm going to switch gear, talk about the bladder cancer. Bladder cancer is very common, and about a quarter of these patients will need to go through radical cystectomy to have bladder removed. The problem with this surgery is it has a high recurrence rate. The mechanism of recurrence is unknown. There's some theory about the tumor spillage during surgery. So we try to see if we can detect any residual tumor cells after the surgery to confirm there was no spillage. We designed this experiment, so we collected pelvic washings at multiple time points through the surgery. So before the surgery, after bladder was removed, after lymph node was removed, and the suction fluid. And for sequencing, we first did the whole exome sequencing to identify all mutations. Then we did the targeted sequencing to reach high coverage in the washings. And in addition to the washings, we also sequenced the blood and the spiking samples for control purpose. So here's the results from the first patient. The spiking confirmed that the results was pretty accurate. When we put in 10%, we measured 10.11% and so on. The blood were all negative. And in the pelvic washing, we did find one sample was positive, and that was after the bladder was removed. And the tumor cell was found to be at 0.27%. And we did this for a total of 17 patients. And after all the quality control, there were 9 patients pass, and these were our final cohort. So in these 9 patients, first, the blood were almost all negative. And for the pelvic washing, before the surgery, they were all negative. However, after surgery, most of them, 7 of 8 cases, became positive. And so at first, it looked like it's negative before and positive after. So seems to suggest a spillage. However, next we noticed if we sort these patients by their peak RCCF, the level of residual tumor cells from high to low, if we sort by this, and we found a striking pattern association with histology. And more specifically, the patient with the highest level of residual tumor cells was the only squamous cell carcinoma. And this was the most aggressive form of bladder cancer, where number 2 to 5 were high-grade urothelial carcinoma with variant histology, so more aggressive. And the rest of 4 were just a plain high-grade urothelial carcinoma. So this was unexpected. And another thing we noticed was for the patients with high residual tumor cells, they were more likely to recur. There was one outlier, but as we look deeper, we found this patient had developed a new tumor, but it was considered as another primary tumor, so it was complicated cases. So we now ask the question, what are we actually detecting here for the residual tumor cells? Initially, we were thinking about the tumor spillage, but that cannot be explained by the association with histology. We now think a more reasonable explanation is before the surgery, there were already some microscopic tumor clones in the surrounding regions. After surgery, they become detectable. That can explain why they are associated with the histology, which is the intrinsic property of the tumor. And we made the cover of the journal. Lastly, I'll briefly touch a breast cancer pathology, tumor contamination in normal. So in tumor mutational detection, we always need control samples. And in one retrospective analysis, we found we have 8 patients of breast cancers where we sequenced the 2 normals. In addition to tumor, we sequenced the adjacent normal and a blood. And if you look, you can see a lot of the mutations in tumor are also detected in the adjacent normal, but none of those were in the blood. So we hypothesized that this could be caused by improper handling during tissue grossing and procurement. And we designed this special collection procedure where you have a tumor and normal tissue. And it depends on whether you collect a normal before you collect the tumor or after the tumor. That's called a PA, or pathologist assistant, clean or dirty. and whether you change your tools each time, that's called tissue procurement, clean or dirty. Combining these two conditions, we have four different conditions. And we did this in 2 patients. And in both patients, the clean was found to have no tumor, but dirty had the highest level of tumor contamination. And the take-home message is, if you have to use adjacent normal tissue as control, try to use a clean collection procedure. So just to summarize, ultra-deep targeted sequencing provides a robust platform to detect the low-frequency mutations. And we have developed 3 applications in different areas. And there could be future applications in cancer prevention and early detection that remain to be explored. I would like to thank all my collaborators and patients. And thank you very much.